My name is Mallory Williamson, and the date is 10/4/2022. Okay, today I'm going to be discussing the senses smell and taste. The first topic we're going to talk about is chemoreception. Chemoreception is a process by which organisms respond to a chemical stimuli that depends primarily on the senses of taste and smell. All senses rely on receptors to transform information about the environment into patterns of action potentials that can be interpreted by the brain. Smell and taste are both forms of chemoreception, which is one of the oldest senses from an evolutionary perspective. Unicellular bacteria use chemoreception to sense their environment, and primitive animals without formalized nervous systems use chemoreception to locate food and mates. It has been hypothesized that chemoreception evolved into chemical synaptic communication in animals. The first sense I'm going to talk about is smell. Olfaction is one of the oldest senses. It allows us to distinguish between millions of different odors. Many animals rely on olfactory cues for survival. Our noses are not as sensitive as animals, but ours do play a major role in everyday lives. We don't really realize how important our sense of smell is until we lose it. I know that when I get a common cold and I lose my sense of smell, it's very frustrating. And some people lose it altogether due to various medical conditions. Okay, so the pathway in the sense of smell. Okay, the modulation of incoming sensory information begins in the olfactory epithelium. And we're talking about this area up in here. Olfactory epithelium lays high within the nasal cavity. This lining of epithelium is embedded with olfactory neurons. The axons of these olfactory neurons form the olfactory nerve, which synapses with the secondary sensory neurons in the olfactory bulb. So this little diagram here is kind of a blown up section of what's going on right here. And so this is the olfactory bulb, and these are the neurons we're talking about. And these are the axons, and these are the dendrites, and this is the cell body. Secondary and higher order neurons project from the olfactory bulb through the olfactory tract to the olfactory nerve. Smell continued. Now we're going to talk about signal conductance. Odorant molecules must first dissolve in and penetrate mucus on the epithelium and basal lamina. So we're talking about down in here. Once this is accomplished, odorant molecules bind to the olfactory receptor proteins on the dendrites of the olfactory neurons. So these are the dendrites. Olfactory receptors are G protein-linked membrane receptors. The combination of most odorant molecules with the olfactory receptors activates a special G protein called GOLF, which in turn increases intracellular cAMP. The increase in cAMP concentration opens cAMP-gated cation channels, which depolarizes the cells. And if the graded receptor potential is strong enough, it triggers an action potential that travels along the sensory neuron axon to the olfactory bulb. So this is the axon. This is the bulb. The axons of neuron cells converge with secondary neurons, these are the secondary neurons, in the olfactory bulb, which then modify the information before sending it to the olfactory cortex. Now we're going to talk about taste. Our sense of taste is closely linked to olfaction. Much of what we call the taste of food is actually the aroma of the food. When you get sick and can't taste anything, it's the aroma of the food you actually cannot smell. It's not that you can't really taste the food due to being congested. Taste is believed to be a combination of 5 sensations. We have sweet, sour, which is acidic, salty, bitter, and umami. Umami is a basic taste that enhances the flavor of foods. It's associated with amino acid glutamate and some nucleotides. It's the reason monosodium glutamate, MSG, is used as a food additive in some countries. So now we'll talk about the pathways. The receptors for taste are located primarily on taste buds, which are clustered together on the surface of the tongue. So this is the surface, the epithelium, epidermis of the tongue, and this is a taste bud. Coffee's up. One taste bud is composed of 50 to 150 taste receptor cells, which can be shortened to TRCs, along the support cells and regenerative basal cells. Taste receptors are also scattered through other regions of the oral cavity, such as the palate. So there's 50 to 150 taste receptors in that taste bud. For a substance to be tasted, it must first be dissolved in the saliva and mucus of the mouth. Dissolved taste ligands then interact with an apical membrane protein, which is a receptor channel, on the taste receptor cell. Interaction of the taste ligand with a membrane protein initiates a signal transduction cascade that ends with release of a chemical messenger molecule from the TRC. Chemical signals released from taste receptor cells activate primary sensory neurons whose axons run through the cranial nerves. So here's the cranial nerves, so the axons will run through them, that area. Lost my spot. Through the cranial nerves 7, 9, and 5 to the medulla where they synapse. Sensory information then passes through the thalamus to the gustatory cortex located in the deep cortical region called the insula. Central processing of sensory information compares the input from multiple taste receptor cells and interprets the taste sensation based on which populations of neurons are responding the strongest. Taste pathways continued. So we're going to talk about signal conductance next. The taste buds contain 4 distinct cell types, 1, 2, 3, and 4, and 4 being the basal cells. So the first one we're going to talk about is glial, which are support cells. The cells responsible for taste, salt taste, have not been definitively identified, but evidence suggests that salt reception may reside on type 1 support cells. So this is the type 1 cell we're talking about. In the current model for salty taste, sodium enters the taste receptor cell through an apical ion channel, which is right here, that sodium channel, which in return depolarizes the cell, setting off a series of events that culminated with the primary sensory neuron firing an action potential. Now we'll move on to the type 2, which is presynaptic cells and taste receptor cells. These cells respond to sweet, bitter, and umami sensations. They express multiple G protein coupled receptors on their surfaces, and these in return activate gustacin, which is a special G protein, which then in turn activates multiple signal transduction pathways. Some of these pathways release calcium from intracellular stores, while others open cation channels and allow calcium to enter the cell. So we're talking about here. ATP. Okay, calcium signals initiate ATP release from the type 2 cells. ATP acts as a paracrine cell on both sensory neurons and neighboring presynaptic cells. This communication between neighboring taste receptor cells creates complex interactions. Now I'll move on to type 3, which is presynaptic cells and taste receptor cells. So these cells respond to sour taste. Increasing hydrogen, which is the sour taste signal, also changes pH. Hydrogen atoms on ion channels of the presynaptic cell from both extracellular and intracellular sides of the membranes. Intracellular pathways remain uncertain. Ultimately, hydrogen mediates depolarization of the presynaptic cell, results in synaptic serotonin released by exocytosis. Serotonin in turn excites the primary sensory neuron. And type 4, which are basal cells. These are believed to be precursors of taste receptor cells. There wasn't a whole lot of information that I found on this type of cell other than that. So I have a couple of diagrams here that are just showing the difference between these G protein coupled receptors, which are associated with sweet, bitter, and umami tastes, and the basic ion channel, which is salty and sour transduction. So all in all, communication between cells and neurons is ultimately very, very important because without proper communication, our body would not function properly.